Hi there, thank you. Thank you, Kevin, for the introduction, and to Warren for organizing this event. I'm from the Malaria Atlas Project. We make pretty maps of where malaria is. And as Kevin said, I'm going to be concentrating on Plasmodium vivax, which has been my focus for the last several years, and how we have been mapping and addressing the challenge of this relatively neglected parasite. And because we have a strict 8 minutes to cover what we need to cover, I'm going to do a flying tour of some of the key points about Plasmodium vivax, malaria biology and epidemiology, why it's considered neglected or how, what challenges we've faced in trying to map this disease, and what we've done to try to meet that challenge. So as Kevin mentioned, there's 5 or 6, if you count the subspecies of ovale, parasites that affect humans for malaria. And the vast majority of the burden or global caseload is attributed to falciparum and vivax malaria. The biology of Plasmodium vivax does impart some unique epidemiological problems that make it difficult to control and even to understand in some ways. As shown here in this life cycle illustration of Plasmodium vivax, you'll notice that there's this liver stage. All Plasmodium parasites do enter the human liver at some point in their life cycle. But ovale and vivax have this ability to stay in the liver and form this dormant phase called the hypnozoite. That stage can then wake up at some point. It could be anywhere from a few weeks to months to years after the initial mosquito bite primary infection, and cause another blood stage infection and then another one after that. And this ability for it to sort of hide out in the liver allows it to overwinter seasonal transmission. It allows it to hide from blood stage treatment, diagnosis. And if we look at a map of what malaria looked like at the turn of the previous century, you see that it extended far into the temperate climate. And that would have been Plasmodium vivax, that light green way up north. And it remains the most geographically widespread of the human malaria parasites. It's still far up in North Korea. And an additional problem of this relapse, so not only does it hide out, but it's difficult to treat that liver stage. There's one currently licensed drug that can treat it, and it comes with its own problems, which could be a whole nother discussion on its own. But it's a long treatment schedule, which people don't like to take a drug for 2 weeks. And it can cause acute hemolysis in people who have an enzyme deficiency called G6PD. So some people are reluctant to give it as a drug. There's other things that Plasmodium vivax does that makes it hard to find. It circulates the blood in low parasite densities. It's often submicroscopic. It's often asymptomatic as well, so people don't necessarily have the classic malaria fever to go for treatment. So we have this widespread parasite. It's hard to understand. It's hard to find. So why would this be one that's considered neglected? And I've been told by a couple reviewers of papers that we can't really call vivax neglected anymore. The fact that I'm even invited to here to talk about it, I think, means that it's gotten some attention. But when we talk about it being neglected, it's relative to falciparum, because as Kevin said, when we talk about malaria, most people think falciparum, Africa. And just to kind of test this hypothesis, I did a search on PubMed of falciparum plus malaria, around 30,000 hits. Then vivax plus malaria, only around 7,000. So it is quite clear that there is more research output for falciparum malaria. And within MAP, our own database that we've assembled of cross-sectional surveys that look for infection in communities, we have Plasmodium vivax here. This is a map of all of our survey points. And we have around 13,000 points, which I was quite proud of because I was, one of the people going around basically begging people for data, and was feeling quite proud of that until I compare it to falciparum, which we have about 42,000 points for. So there's a huge discrepancy even within our own group where we use the same methods for both parasites. that we don't have as much evidence to support what we try to do for both. So given what we have available and what we know about these parasites, what could we do in terms of trying to map it and understand how differences in the spatial distribution of this? So what we started doing at MAP was the first thing we did several years ago was we mapped the limits, the geographic limits of transmission, and that allowed us to assess what the population at risk is worldwide. And then within that, we estimated the prevalence using those survey points I showed on the previous slide. But what we're now trying to do is generate an estimate of how many new clinical cases happen each year, which is a very different metric, but we think is more operationally useful for people who work within country programs. So in order to go from prevalence to an incidence map, we first needed to understand, does relapse vary geographically? So this phenomenon of when the hypnozoite wakes up. Because there's been a long-standing hypothesis that the tropical strains will wake up quite quickly after the primary infection, whereas the temperate strains wake up more slowly so that they last through the non-transmission season. And so we did a large analysis and found that there are indeed geographic zones of relapse. So we developed a mathematical model to relate prevalence, which we had mapped, to incidence, which we want to map, and we had separate model fits for those different geographic zones of relapse, assuming that relapse would affect both prevalence and incidence measures. But our models and our outputs are only as good as the data that we have to put into that. So if you look on the previous slide, some of our relapse zones have almost no data, and some of the zones had no data. And I was fitting this to about 175 points. And these 13,000 points that I have as our evidence to predict prevalence, you can see that they're not homogeneously spread out throughout the vivax endemic world. We're heavily biased to areas that have national household surveys like DHS and MIS, which don't commonly search for vivax, and from areas where we have support from other Oxford institutions where we have strong collaboration. So by far, most of our data is from Indonesia, thanks to our colleagues based at the Eichmann Oxford unit. So what can we do to meet this challenge? We don't have a lot of data. We have a complicated parasite. And what we want to do is capitalize on the type of data that is strongest from different regions of the vivax endemic world. So in the Americas, they have strong, relatively strong health systems, health information systems. So we could use incidence data that is reported from health facilities to the government to measure incidence. And in other areas where we know that the government isn't reporting any vivax, like in Africa, we can use this modeled relationship that we have. And the outcome is that we have a hybrid map that utilizes health information system data, which we of course adjust for things like reporting completeness and treatment seeking, because we know that even in strong health information systems, not all the facilities report, and not everyone who gets sick goes for treatment. But even with those sort of flaws that we have to adjust for, we still think that an adjusted health information system database is better than a model that has a weak evidence base. As things progress towards elimination and strong control, the irony is that the less malaria an area has, the better that they are at finding their malaria cases. And we're finding that in some elimination, the countries that have already set elimination goals, that they're able to find, record, and geoposition cases better, such that we can make more fine-scale resolution maps and allow for malaria stratification for control and elimination subnationally. We only have a handful of countries, but I'm very optimistic that this is going to improve even further over the next 15 years, as Kevin showed with the funding slide. So that's a very brief overview of what we've done with vivax and MAP, and I want to thank our whole team that's contributed in different ways, and our funders, the Wellcome Trust and Bill and Melinda Gates Foundation.